Hello and welcome back to The New Creationist. I'm Eugene. Today we're going to be debunking the homology argument for evolution by looking at the genetic evidence. There's actually quite a few genes that contradict evolutionary common descent. Today we're going to be looking at three of them. The first gene we're going to, the first one is FUB-P1, the second one is WNT5A, and the third one is CIC. Let's take a quick look at the first one, FUB-P1, which is short for the far upstream element fuse binding protein 1. We're going to come down and look at the ortholog list here in the Ensemble Genome Browser. And first, before we even look, I'm going to click primates here under the show details heading. The first thing you want to do is first think about what evolution would predict if we have a most recent common ancestor with chimpanzee. What would evolution predict? What species should the human FUB-P1 gene be most similar to? Chimpanzee, right? Well, let's take a look. I'm going to scroll down. Actually, first let's look at all the different primates, because I have the primates button already clicked. The primates for the FUB-P1 gene that show homology from the greatest percentage of similarity to the least. First macaque, next marmoset, after that gorilla, next chimpanzee, after that bush baby, gibbon, tarsier at 93%, mouse lemur at 86%, orangutan at 75%. Notice these are all one-to-one comparisons. Okay, now, of course, the first question is going to be, why isn't chimpanzee on the top of the list? Marmoset, then gorilla. Now it's rather peculiar, and we could go and actually let's go look really quickly at the cDNA alignment for marmoset, just to see what. It's going to take a second here to load. And what this basically does is gives you the alignment of the nucleotide sequence for this particular gene. And you can see in the human on top is the top line here is the human sequence. That there's a big indel here. And there's a few substitutions here and here, here, etc. Here, another indel here, and so forth. And it's actually a fairly large gene, so you've got quite a few nucleotides in the exon sequence. Now, let's go back and look at another primate. The next one, well, actually, it went back when I clicked back. It unchecked this box. So the original question: what species should the human FUB-P1 gene be most similar to? And the answer is, amazingly enough, hold on, is it panda? Panda. Okay, the target ID is 96%. For marmoset it's 97%, but the actual query percent ID is 100%. The human gene as compared. So when you look, when you compare from human to panda, 100% of the human sequence is in panda. but the target ID is lower, 96%, most likely because of indels, just like we saw the indels in the marmoset sequence. Of course, the main question is, why then would the human FUBP1 gene be more similar to panda or marmoset or dog and cat? Here, even you've got cat being at 100% with a 99% target ID. So the query percent for in gorilla at 100% target, 100% query and 97% target ID, and chimpanzee 100% query and 97% target. Why is a macaque then more similar? Why is well, gorilla is about the same. Of course, we could go down in the alignment and actually count nucleotide differences. But here the percentage for cat is clearly greater, a greater percent of similarity to human than chimpanzee is. Very peculiar. So why then would the human FUBP1 gene be more similar to cat than chimpanzee? Are we saying now, in other words, if a 98% similarity rate between human and even if it were true that humans and chimps were 98.5% similar, why would that be evidence for common descent? Well, simple. Because if we do have a most recent common ancestor, we should be about 98.5% similar because of the slow mutation rate. So if DNA mutates at a fairly constant rate, even from generation to generation, even though there seems to be a lot of disagreement as to exactly what that rate is, and the measured rate is much slower than the predicted rate based on both phylogeny. In other words, if we were to assume that there is a common ancestor, then the rate would be about between 130 mutations per generation and 160. However, the rate is, according to the last study I saw, much less, about half that, maybe even less. Somewhere between 30 mutations and 60, if I remember correctly. And we can actually, there's some studies done, variation in genome-wide mutation rates within and between families, human families, and they go through certain. Of course, this is a paid site, and you'd have to actually pay to read the article. So I'm not going to be doing that right now. In fact, I can't afford to do it anyway. So if you wanted to go ahead and spring for it, let me know what you find. However, just looking at the DNA sequences, which are posted online for free, if we were to go through and check the alignment for cat with human and go through and count the number of differences, here you get a few substitutions, the percent identity at 100% actually means there's not enough substitutions to actually add up to more than 1%. So it kind of rounds up to 100. You've got quite a few substitutions here, but not enough considering the size of the gene. And you have here, it looks like one indel at the end. So again, we basically just come down to this simple question. Why is a cat more similar to chimpanzee? Let's go back and look at the chimpanzee sequence. See if we can find it again. I'm gonna go ahead and click on show details real quick. So I mean for primates, so I can get right to chimp, and let's compare that DNA sequence. I'm just going to browse down it again. Huge indel here. Another indel here. And other than that, and not so many substitutions, but those two indels reduce the amount of similarity quite a bit. And that's why it's so you've got here a peptide length of 644 amino acids in human, but there's an additional 21 amino acids in chimpanzee. So the actual protein length for chimpanzee is 665 amino acids long, which is a considerable chunk of DNA. The next gene, WNT5A, I've already got the primates node clicked here, so we're going to go down and look at all the primates. Again, here we've got chimpanzee at the bottom of the primates when it should be at the top, since we're comparing human to the others. And we even have tarsier, which is more similar to human than chimp. Mouse lemur at 99 and 99. So the percent of similarity between mouse lemur and human is considerable compared to chimpanzee. At the top, marmoset is more similar to human. Gorilla. Let's see how we get marmoset, gorilla, mouse lemur, bush baby, orangutan. So why then is chimpanzee at the bottom of the list for primates? Now let's go ahead and uncheck this box and look at other species. Marmoset, gorilla, dog. Okay, so we got a dog here now. Actually, this time let's check the protein alignment. Just because of the size of the gene, sometimes it's easier to go look at the protein sequence because there's fewer, there's less to look at, since 3 nucleotides make an amino acid. So in the sequence here, you've got less to look at, about 1/3 less. So here we've got some substitutions. Not many. So for dog, the similarity between dog at 380 amino acids each for human and Canis lupus familiaris, 99% similarity, where for chimpanzee, let's go back, second to load. Now go look at chimpanzee. We know it's at the bottom of the list. We're going to click that and go down, and protein alignment, second to load. And here we go. Let's see. Okay. Another huge indel, some substitutions, and more substitutions up here. So we have a considerable difference between human and chimpanzee, and 380 amino acids. And that just blows the doors off. How in the world the human DNA sequence be more similar to dog than chimpanzee? Alright, now the last gene, CIC, we're going to have a similar, actually we've got one with no ortholog. Let's see which one that is. Go ahead and click on primates. And what would evolution predict? Alright, well, mouse lemur is here at 49%, so we're just going to take a mental note of that first. What's not here? Tarsier? Okay, Tarsier is not in here. Of course, Tarsier is a primate, so immediately we have a flag off in our head. Now, I could go through and just manually do a blast of Tarsier to see if maybe it just got left off the list. Maybe the sequence for Tarsier in that particular region isn't complete. I'm not really going to worry about it right now. Right now, we have chimpanzee. Oh, wait a minute. We got chimpanzee at the top of the list. Exactly what evolution would predict. Okay, so that's fine. But of course, the previous one, chimpanzee, was at the bottom of the list. Here we have chimpanzee at the top, where it should be. However, and gorilla is next, and then orangutan. Okay, but let's go back and uncheck this primate. What we're thinking about is another primate at the bottom of the list at 49% is going to be less similar to human than dog. Again, we have the dog problem. Kangaroo rat, cat is still more similar to human, and squirrel is more similar to human. Ferret, rabbit, and pig, cow, more similar to human. Guinea pig, a horse, mouse, and rat, more similar to human than one of our beloved primates here, mouse lemur. Even a hedgehog is more similar. Wallaby is more similar. Now, a mouse lemur is a primate. It's a lemur. Come on. Okay. Well, there's the three genes that totally throw evolution for a loop. Because what would evolution predict? What should be more similar to human genetically than a chimpanzee? And yet we have problems. Thank you very much. That's going to be all for today, and I hope you have a wonderful week. I'll try to get back to you as soon as possible.